IBESS, Investigating Ecosystems Part 2, is the second of two movies covering topic 2.5 in the ESS syllabus. These two movies focus on the techniques for measuring ecological components, both living and non-living. Part 2, this movie, focuses on the following: measuring biomass, the Lincoln Index for estimating the size of animal populations, Simpson's diversity index, and the use of a biotic index. The essential ideas are: the description and investigation of ecosystems allows for comparisons to be made between different ecosystems and for them to be monitored, modeled, and evaluated over time, measuring both natural change and human impacts. Secondly, ecosystems can be better understood through the investigation and quantification of their components. Here is the outline of the two movies available in the Topic 2.5 Investigating Ecosystems series. Use this outline to find the movie you need for review. This is Part 2, and it examines measuring biomass, the Lincoln Index, Simpson's diversity index, and a biotic index. Here is the first IB syllabus statement of the movie: methods for estimating the biomass and energy of trophic levels in a community include the measurement of dry mass, controlled combustion, and extrapolation from samples. Data from these methods can be used to construct ecological pyramids. In the first semester of this course, we had the experience of running an experiment measuring NPP as a function of different nitrate concentrations. But we also measured biomass of the producer trophic level. I'm hoping you remember. We harvested the grass from small 400 centimeter square plots. We dried the grass in an oven at low temperature for over 48 hours, and then mashed the grass using a balance. We calculated the averages of biomass, grams per square centimeter. If we were interested in the biomass over a larger area, we could have extrapolated the producer trophic level data by multiplying up. Biomass of the producer trophic level can be done by collecting plant samples from a known area in the field. Dry the plants in an oven. We do not burn them, we just dry them. We mass them and then calculate averages of dry mass, grams per centimeter square, and then extrapolate to generate biomass data for the producer trophic level in a larger area. Removing the water by oven drying is important because water is not organic matter, thus not seen as a part of an organism's biomass or growth. Water is also highly variable within the body of an organism, hour to hour and day to day. You should remember the investigation that we conducted measuring the NSP of a snail. Not unlike the NPP grassland experiment, we could have masked the snail to get biomass data for a consumer trophic level. This can be done. Collect the animals from a known area in the field to generate a mass. But would we have to kill the snail in order to ensure that our data was dry mass? Ethical issues arise. Consumers can be trapped in a known area in the field and masked, but this is a wet mass. Evaluate. You need to be able to evaluate the method. Dry mass best represents biomass of the trophic level, but to measure the dry mass of a consumer would raise ethical questions. The water within organisms is not organic matter, thus wet mass of an organism is not representative of true biomass. Water within organisms is highly variable, thus an accurate representation of biomass requires that the sample organism be dried in an oven, seen here to the right. For consumer trophic levels, drying the samples presents ethical issues. Data from measuring biomass or energy can be used to construct ecological pyramids. On the left is a biomass pyramid from a terrestrial system. Remember that unlike energy pyramids, biomass is a measure of a standing stock, simply grams per area. Biomass pyramids are not constructed using rate data. On the right is the biomass pyramid for an aquatic system. Remember that in aquatic systems, the biomass pyramid can appear to be inverted because phytoplankton reproduce so quickly. Thus, the standing stock of the producer biomass at any one instant in time can be less than the primary consumer biomass. Keep this in mind. This should be a review from what you've studied earlier. Now, you need to know a method for estimating the energy of trophic levels in a community using controlled combustion. A device known as a calorimeter burns a sample of organic matter, and the energy per unit area per unit time can be measured, and data from the controlled combustion work can be used to construct energy pyramids. Data from controlled combustion of organic matter in a calorimeter can be used to construct energy pyramids, as you can see here. Remember that the second law of thermodynamics is the explanation for the successive decline in available energy from one trophic level to the next. And all energy pyramids have the same shape. They can have no other shape. Be thoughtful. This should be review. All energy pyramids have the same pyramid shape, displaying the successive decline in the available energy with each trophic level. This is the second law of thermodynamics at work. You should remember the general 10% rule of energy efficiency as energy transfers from one trophic level to the next. In this diagram, the units are incorrectly displayed as joules. The units for an energy pyramid should be joules per area per time. It's a rate. Now let's evaluate methods for estimating biomass at different trophic levels in an ecosystem. Sample locations should be selected randomly. Record mass data and area data from many samples. Ensure that the data is dry data. Water must be removed by an oven. Calculate averages. Samples are taken over the entire year over time to account for the different seasons, and then multiply up, extrapolate to estimate values for the larger area. Evaluation requires examining the specific situation. Is it a grassland or a forest or an aquatic ecosystem, plant or animal that is being sampled? Pay close attention. I've been through some of the general strengths and limitations before, but it's important for me to note some of the differences. Biomass data requires harvesting the plant or the animal. Grasses grow back quickly, but forests do not, and are animals harmed in the process? Remember that dry mass is more accurate than wet mass. It's okay to dry plants, but the dry mass data with animals involves the death of the animal, and this raises ethical questions. Now let's move to a new IB syllabus statement. Direct and indirect methods for estimating the abundance of motile organisms can be described and evaluated. Direct methods include actual counts and percent cover. Indirect methods include the use of capture, mark-recapture with the application of the Lincoln Index. Now, just to be clear, the data collected is the number of animals caught in the first capture, the number caught in the second capture or the recapture, and the number caught in the second capture that were marked. All of this data is used to calculate the population size estimate. It goes like this. Traps are placed in the ecosystem to capture animals. Captured animals will be marked and released back to the general population. Animals are captured and then marked, then released to the general population. Pitfall traps could be used to capture insects, but captured insects must remain alive upon capture and must be released alive with the potential to be recaptured. Again, animals in a known area are captured and marked and released to mix with the general population. Here is an animal that has been marked upon having been captured a first time, but it is critical that the marking not influence the survival of the animal. The animal cannot be made more visible to predators by virtue of the marking. If so, the collected data is not reliable. As you could see in the last image, the animal had numerous marks. A strategy for numbering captured animals can be seen here. A mark here is animal 1. A mark here is animal 2. A mark here is animal 4. But a mark here and here is animal 3. A mark here and here is animal 5. A mark here and here is animal 6, and so on. Remember, the marking of the animal cannot influence the survival of the animal, otherwise the collected data is not reliable. Remember, the mark cannot be brightly colored or too heavy, such that it might influence survival of the animal. This slide shows various animals being marked after the first capture prior to their release back to the general population. And then, once marked, animals are released. They must have time to mix thoroughly with the general population before the recapture phase is undertaken by the scientist. So here is an example, and let's see if you can calculate the estimated population size. Capture 25 fish in a small pond. Mark the fish by putting a notch in the fin. Release the fish. The marked fish need time to remix with the main population. 2 days later, after the marked fish have mixed thoroughly with the general population, 35 fish are recaptured. 14 of the 35 fish in the recapture are marked. 14 are marked in the recapture. Calculate an estimate of the population size. Remember that N1 is the number caught in the first sample, N2 is the number caught in the second sample, and NM is the number caught in the second sample that were marked. So 25 fish are captured, marked, released, 35 fish are recaptured, and 14 fish of the recapture were marked. Now I think the absolutely best way of remembering the formula is to think of it as a proportion. The left side of the proportion is the first capture, with T being the estimated size of the population. The right side is the second capture data. Number marked in the second capture divided by the total number in the second capture. So, if we follow the algebra, our proportion is rearranged to this equation and then rearranged to this equation. Now, I need to note that this is the equation that is frequently given, but I think this equation is more difficult to understand and remember than the proportion that I have given you at the top. Now, if we plug in the numbers, T is estimated at 63 animals, 63 fish in the general population. Now, you need to be able to evaluate the Lincoln-Peterson index. Let's think carefully through the evaluative statements on this slide. First, quantitative measurements of motile organisms help us characterize ecosystems. This is positive. The population should be nesting, remaining in the area sampled, not migrating. The food in the trap must be attractive to the animals. The mark cannot influence survival. We must assume that the marked animals mix evenly with the main population, and they need to be given enough time to do so after they've been released. We have to assume that there are no births, no deaths between the first capture and the recapture. The capture process itself, the use of the trap, cannot influence the possibility of recapture. And you must ensure a large sample size, but time and money are always limiting. So that brings us to a new IB syllabus statement. Species richness is the number of species in a community, and it is a useful comparative measure. Diversity index is a function of both the number of species, the richness, and their relative abundance. But keep separate the diversity index from the general definition of biodiversity, which is the variability amongst living organisms from all sources and the ecological complexes of which they are a part. This includes diversity within a species, between species, and among ecosystems. Let me start the discussion on diversity indices by asking which of these two communities is more diverse, and can you explain your response with reference to the number of different species, the richness, and their relative proportions. Hopefully you would say that the top system is more diverse because there are more different species, 1, 2, 3, and 4, whereas the bottom system only has 1, 2, and 3. Now, which of these two systems would be more diverse? Look carefully and notice that each system has the same richness, the same number of different species at 4. But the top system would be considered more diverse because the relative abundance of each species is more even. In other words, the bottom system is less diverse because it is dominated by one species. So remember, the diversity index is dependent on the number of different species, which is the richness, the relative proportions of the species compared to each other. So let's move to the calculation of Simpson's diversity index. So, students are expected to be able to apply and evaluate the result of the Simpson diversity index. Using this formula, you'll see it on the next slides. The higher the result, the greater the species diversity. This indication of diversity is only useful when comparing two similar habitats or the same habitat over time. D is the Simpson diversity index. N is the total number of organisms of all species that are found. Small n, lowercase n, is the number of individuals of a particular species. The calculation of Simpson's diversity index can be handled in two ways depending on how the data is collected. If the data is collected as counts of individuals, here is the formula. Now remember, you just have to be able to use the formula, not remember it. N is the total number of individuals of all species found, whereas lowercase n is the number of individuals per species. So diversity is equal to the total number of individuals of all species found times 1 less that number, divided by the sum of number of individuals per species times 1 less that number. If the data is collected as percent, not counts, then Simpson's diversity index calculation is this one, where diversity is equal to 1 divided by the sum of the percent of each individual species squared. Let's look at some examples and practice the calculation. We have 4 communities with the potential of 5 species. Location 1 has only 4 species equally represented. 1 divided by 0.25 squared plus 0.25 squared plus 0.25 squared plus 0.25 squared gives us a diversity index of 4. Practice the calculation using the formula on the previous slide, percent data, and work at it until you get 4. But the second location has 5 species, so you would expect its diversity index to be higher, and this makes sense. But the third location also has 5 species, but not quite as evenly distributed, as one of the 5 is represented in fairly low percentages. So the diversity index value is lower than in location 2. Now, the fourth location also has 5 species, but the relative abundance is quite uneven as the system is dominated by species A. Thus, the diversity index is quite low, the lowest of all. You can see the importance of relative abundance in the calculation of a diversity index. Here's a chance to practice. On this slide, we see 3 locations, site X, site Y, and site Z. We can see that site X has far more different species than Y or Z. Thus, the species richness of site X is higher. Thus, its diversity index value is also high. But sites Y and Z each have the same number of different species at 7. Notice that the relative abundance differs between the two sites. Site Z has a more even distribution, whereas site Y really appears to be dominated by one species. Thus, the diversity index of site Z is higher than that of site Y. On this table, data are presented from different altitudes in the Himalayas, from low altitude to high altitude. Notice that the number of different species goes down as you go up in altitude, but evenness actually goes up slightly. Nevertheless, in the Shannon Diversity Index, species richness is the more important variable, so diversity goes down as you go up in altitude. And lastly here, you should be able to draw graphs as seen here to illustrate species diversity in a community over time or between communities. You should remember our study of succession. Diversity increases as succession proceeds over time. More structural heterogeneity, more niches, results in higher diversity. And that brings us to these IB syllabus statements, the last of this movie. Some species can be indicative of polluted waters and can be used as indicator species. A biotic index indirectly measures pollution by assaying the impact on species within the community according to their tolerance, diversity, and relative abundance. And lastly, explain the use of indicator species and biotic indices in measuring aquatic pollution. The presence or absence of certain aquatic species can be an indirect indicator of water cleanliness. Let's take a look. The presence of certain species in an aquatic system, like these tubifex worms in the upper left corner, is an indication of pollution. And the presence of other species in an aquatic system, such as the stonefly, is an indication of clean water. Walking into a stream with a net and a certain knowledge of data collection allows one to collect aquatic species for examination in order to understand water quality. Here's the method. The kick sampling method for assaying species living in a river ecosystem involves placing the net downstream of one's feet and with a standardized motion disrupting the stream bottom. Invertebrates, insects and worms living on the stream bottom can be collected as the current carries the invertebrates into the net. The species seen in these photos are indicative of clean waters. These species have low tolerance for polluted waters. Indicator species are an indirect measure of water cleanliness. The species seen in these photos are indicative of polluted waters. These species have high tolerance for pollution. Indicator species are an indirect measure of water pollution. The presence or absence of certain species can be quantified or placed on a scale to determine a biotic index. A biotic index indirectly measures pollution by assaying the impact on species within the community according to their tolerance, diversity, and relative abundance. Pay close attention through the next few slides. The presence or absence of certain species can be quantified or placed on a scale to determine a biotic index. Species can be given a tolerance rating that can be used to indirectly assess pollution. Here is a data collection document used for the purposes of determining a biotic index. You can see the tolerance rating here in these four groups. One can sum the number of individuals of each species, then multiply by a pollution tolerance rating, then divide by the total number of individuals in the sample to provide an index, a biotic index. The index can be used to assess water cleanliness. Low values indicate clean water. High values indicate polluted water. This table displays data from various locations. The data includes the number of fish caught, the number of different species richness, the percent of fish tolerant of low oxygen conditions, low oxygen is typical of polluted conditions, and the percent of fish tolerant of disturbed conditions. A biotic index score is mathematically calculated, and according to the biotic index, the water is given a rating. Some species can be indicative of polluted waters and be used as indicator species. A biotic index indirectly measures pollution by assaying the impact on species within the community according to their tolerance for the pollution, their diversity, and relative abundance. Can you explain the use of indicator species and biotic indices in measuring aquatic pollution? Here is a nice image displaying pollution released as industrial discharge and effluent from a specific point in a river, and the current carries the pollution downstream. From the point of pollution, you can see an increase in the pollution-tolerant species downstream. From the point of pollution, you can see the oxygen levels decline in the river. But the further you are away from the discharge, the pollution-tolerant species disappear as the clean water species reappear, and oxygen levels also rise. And that brings us to the end of Topic 2.5, Investigating Ecosystems, Part 2.